I'm Stephanie Smith, and I'm the co-founder of Beagle Bioproducts, and I'm really pleased to be here today with Wayne Carmichael.
Thank you.
Wayne has been a mentor and friend of mine for a long time, since we worked together at Wright State University. And I just wanted to talk to Wayne about his, is it 35 years of experience in harmful algal bloom work?
We'll admit to 40.
Okay, okay. So starting this type of work 40 years ago, tell me how you first became interested in harmful algal blooms and this type of work.
Well, that will date me, but on the other hand, it is important to start at the beginning. Obviously, cyanobacteria are an old organism, but the recognition that they might be producing harmful toxins really wasn't recognized as coming from cyanobacteria until the 1950s and 1960s. And my mentor, who was Paul Gorham, who worked originally at Caltech and actually developed some of the original culture media for algae, moved to National Research Council in Ottawa, even during the Second World War, because he was actually employed by the Canadian government to develop hydroponics, because at the time of the Second World War it was important to perhaps grow hydroponics because of all of the needs related to products in the Second World War. And out of that, he also had an interest in water and water quality and algae, and began looking at blooms of algae in the prairie provinces of Canada to answer questions about intermittent but repeated poisonings of animals from ponds and lakes in the prairie provinces, even in areas of Ontario as well. And so he was the first using that ability to culture algae to take some of the algae from these blooms associated with what appeared to be animal poisonings, and then go into the lab and grow those algae and show that they produced some sort of a toxin. So at the point I entered the picture in 1970, he had left to become head of the botany department at the University of Alberta, and had brought with him some of these key original type cultures from Saskatchewan primarily. And those cultures were important because he had already established that they were producing something that would kill animals. And of course these were all at that point test animals in the laboratory, mice and primarily. But they had come from situations where cows and sheep and horses have died. So it sort of combined my interest. I had come out of a botany department at Oregon State University and had an interest in phycology, not necessarily toxic algae, but I had an interest in phycology. He provided me the opportunity to work with algae and see if we could come up with some sort of way to study the toxic compounds that these were producing. So what we were left with in 1970 was the ability to grow these algae. And the second thing that was important, we were placed in the middle of the prairie provinces, which was probably one of the best places that you could have been in North America to get opportunities to collect bloom materials from these prairie lakes. So I set about two steps. Initially was just to collect from different poisoning episodes, identify and culture, and from that culture material use the cells to at least do animal studies, animal toxicology. And the interesting thing that came out of that was that the cultures that he had seemed to be producing two different toxins, a neurotoxin and what we now know as a liver toxin. But what we found collecting from different lake sources was that there was, while that seemed to be the key, there were neurotoxicities and hepatotoxicities, there were sort of subgroups within that. So by the time I finished my PhD work, I was convinced that we had more than two toxins, that we had maybe 4 or 5 different toxins. So that for my postdoc work, I then made contact with a pharmacologist in a pharmacology department to work with first Anabina and its neurotoxin and try and determine its mechanism of action. We already knew what the structure was, Paul had determined that structure with a group at in Ottawa. So we had the structure. We knew it was an alkaloid, and alkaloids are often neurotoxic, but we didn't know what the mechanism was. So working with small animals and animal tissue preps, I did standard pharmacological methodologies of dose and dose response and blockers and enhancements and neurotransmission to come up with a mechanism of action for anatoxin A, which is what we then named this particular toxin. So that, having had that further convinced me that we had a toxin and a mechanism of action and a symptom that went with that in animals, but we had all these other symptoms as well. So when I took cultures from Canada and moved to Ohio, Wright State University in 1976, I set about systematically working with individual organisms to look at their toxic components, isolate, purify, and characterize both chemically and toxicologically.